about Rumen fungi. The introduction. The fungal kingdom is one of the oldest and largest clades of living organisms, and it includes phyla, um, Ascomycota, Basidiomycota, Chytridiomycota, Plasticladiomycota, Glomeromycota, and Zygomycota. Here there is in herbivores three parts of stomach. One of them is rumen, and inside the rumen there is chytrid that decompose the cellulase. Mycota are monophyletic groups with different branched clades and zoosporic fungi, morphologically the only fungal phylum that reproduce by monoflagellated zoospores. Exception Mycota are multiflagellated. Chytrid are simple and there acarbic chytrids, and there is halocarbic chytrids. Zoosporangia, and there is resting sporangia depend sexual and asexual reproduction. In zoosporangia, the wall is thin, and in resting sporangia is thin. Here is eucarbic fungi. The common chytrid. The common chytrid is most unusual chytrids are the obligate and Arabic Mycota, which grow in the rumen of herbivorous animals. Chytrids convert the inorganic compound into organic compound, and anaerobic chytrids occur in the rumen of many larger mammalian herbivores. A new column Mycota in comically important obligate anaerobic chytrids. And they have hydrogenosomes to generate enzymes growing in the herbivores, nucleomystics, and decompose cellulose, hemicellulose, and break the lignin deposits into smaller pieces and allow surface of bacteria to decompose lignin. They have a main part in the microbial consortium of the rumen environment. They have part in carbon metabolism depend on obligate anaerobic fermentation of glucose to acetate, lactate, ethanol, and hydrogen. Anaerobic chytrids are monocentric or polycentric zoospore and flagellated or multiflagellated insects on a plant. Anaerobic aquatic phycomycetes occur in the rumen, cattle, sheep, and goats. Suggestion as significant roles in rumen function. The spore of the fungi colonize and invade fibrous plant materials and are involved in cell wall polysaccharide degradation and fiber digestion. Anaerobic fungi have been cultured on a wide range of polysaccharide, including cellulose, starch, xylene, and other hemicellulose, indicating that they produce the range of the enzymes necessary to degrade these polymers, simple sugar. Isolate nucleomystics produce extracellular cellulase, carboxymethylene cellulase, and xylenase. However, investigation have been mainly focused on nucleomystic volatiles. The properties of other fungi have not been studied, showing that nucleomystic party serum and an undefined isolate have produced a wide range of glycogen. Three anaerobic fungi, frontinase and S communase and P communase, in order to determine the contribution of these fungi to rumen function enzyme production by rumen anaerobic fungi grown on sessile fiber when they were grown in the presence of sessile fibers. And Von tells S communase and Pheromonas communase secreted into the medium several enzymes as the maximal activities and maximal growth of culture occurred at the same time. Analyses were done on 6 days. Old cultures and from sales had a highest P-1 for glucanase activity, while the two other fungi showed greater exoenzyme activities. Among the exoenzymes, the galactose had a very low activity, 16 to 30 times lower than of P-fucidase. In order to evaluate the role of these fungi in fiber digestion, the natural and these three species were determined when the culture in present sessile and fontaine and produce B-glucanase and glucosidase and galactose as a previously and there is experiment to improve that and many reports of that in uh, related to our as most studies have focused on physiology of a nucleomystic species, it was interesting to determine and to compare hydrolytic activities produced by two other species, P. communis and S. communis. They exhibited the same range and natural of zero and endo acting enzyme produced by N. frontales. They were mainly secreted and their activities were of the same magnitude when the fungi were grown in the present of Caesar. The enzymatic properties were similar from one enzyme to another and one species to another. These properties cannot be used for the identification of a new isolate. The enzymes accepted the same optimum pH 6 to 0 and optimum temperature of 50 Celsius except for P oxidase. These values are similar to those for xylanase and glucanase from different isolates of nucleomystic, but different from those of enzyme degrading official and a wide range of enzymes produced by nucleomystic Batricium and B. communis, which were assayed at 39 Celsius. The properties of the enzyme were different to those described for enzymes of aerobic fungi, which are commonly active at more acidic pH value from pH 4 to 5 and higher temperature from 55 to 7 Celsius. Particularly the p-oxidase were unstable in the absence of substrate after relatively short incubation 4 hours at 50 similar Celsius. At 50 Celsius. Similar results have been reported for B-glycanase and xylanase from different nucleomystics isolates were similar to those of the enzymes of three different isolates studied by Williams and O'Brien. The role of Comenius in the Rumen was not clear as these species have a very limited rhizoidal system compared to frontales and Pyromonas communis. Our results show that the S. communis secrete the same wide range of glycosides hydrolysis produced by nucleomystic fronta and is able to degrade cellulose and hemicellulose and non-cellulosic mixed glucans of plant cell wall. This, although as communist does not penetrate plant tissue, it probably acts as an active degrade. by secreting large amount of extracellular. Those our reference, and thanks for listening me, Abrar, and my group that's for this presentation, Fatma Ali, Hiba Barak, and Sarah.